Hello, welcome to the new year of DevoWorm. I don't see anyone here yet, so what I'm going to do is I'm going to get started, and I'm going to talk about a couple things today. There we go. Hello, Ujjwal. How are you? Oh, good. Okay, so Ujjwal's here. So I wanted to start off, first of all, and I'll share my screen here. I'll start off by saying that Ujjwal and I have this preprint out now. This is Periodicity in the Embryo, Emergence of Order in Space, Diffusion of Order in Time. And this is a paper that we've talked about over the past several meetings in the previous year. We have now submitted it to a special issue of Biosystems, but we also have the preprint up here as an open access version. So we have a couple of what the metrics are on this. So we've got 282 abstract reads since about Friday. I think it was accepted on Friday. So it's, it looks like people are kind of interested in it. I already tweeted about it, and so it's, it's out there now. And so yeah, so just an update on that. How are you doing today, Ujjwal? Are you having a good new year so far? Okay. Very good. Yeah. Oh, good. Things are good. Well, glad to hear that. Before today we're going to talk about sort of a little bit of a plan for 2021. I don't really have a full plan yet, but I wanted to go over some things and talk about things that we did in the past year, and then how that might relate to what we do this year. So I was going to kind of wait for a couple more people to show up, but I think I can start now. And then if you're watching this on YouTube, okay, just finished off your aspect detection paper. So this was a machine learning paper that you were doing. NLP, okay. Well, very good, yeah. It's nice to see. It's always fun to have papers be completed and released into the wild, or at least submitted somewhere. So yeah, that's good. So again, we're gonna I think we're gonna start with the presentation here, and this again is just kind of growing into 2021. This is going to, let me share my screen. This is more or less a recap of 2020 and where we might be going for 2021. So this is growing into 2021. You can see that we did this last year where I had this little baby worm representing the new year. And so there are a lot of things we could do this year. In fact, last year I was going over the presentation for last year, and I found that a lot of the things that we talked about doing for this year we kind of did, but didn't do it exactly in the way it was planned. But which is fine, and we did a lot of things that we hadn't planned on doing, which is also fine. It's actually good because when opportunities arise, it means you can take advantage of them. So we have our new logo here in the top right-hand corner, and I will present this, put this in present mode. So growing into 2021. So this is a quote I got out of an article called On Dinosaur Time from the Smithsonian Magazine. And it was an article on paleontology and sort of the era of dinosaurs versus like the time, all of geologic time. And one of the lessons you can learn from this paper is that less time separates Homo sapiens from Tyrannosaurus rex than separated T. rex from Stegosaurus. Meaning that the Tyrannosaurus rex, which appeared later in the era of dinosaurs, is closer to us today than T-Rex was from Stegosaurus, or a dinosaur that appeared early in the dinosaur era. So I like to, every time I give a timeline, I like to give a quote like that to put the flow of time in perspective. And so thinking about like how things happen in sequence, I think it's enlightening. But for our purposes, we have 2020, which may have seemed a lot longer than it was. And I have this timeline that I've prepared. So what did we do in 2020 in terms of our meetings? We actually did a lot. I have a spreadsheet of meeting recordings, meeting topics, and dates that are available from the website. If you go to the Weebly site for DevoWorm, you go to the the final tab. It's like how to get involved, and then you can click on to the list of meetings for 2020, and so all those meetings are listed, and there are a lot of really interesting topics. But two of the things we did like in February and March. One was a talk by Tom Porteges, who's a member of the group, a long-standing member of the group. He attends the meetings occasionally on something called bio-inspired modeling. And that was largely on a lot of the work that he was doing. He's done a lot of work with something called Morphozoic. He's also done some work on something called Morphognostic, which is a sort of the Morphozoic project that he's working on involves like morphogenesis. Morphognostic involves more like sort of cognitive stuff. And so currently I think he's modeling the behavior of honeybees. He's also interested in nest building in fishes and other types of things like that. So he's got a lot of interesting approaches to a lot of interesting problems, and we had him give a talk about that last February. We also had a talk from George Mikhailovsky, who's a researcher who has a background in physics and biophysics, and he did a talk on structuredness and entropy, which is also something that if you go back to the list, you'll see it on the list in March. And I encourage you to watch that video if you haven't already. It's based on a paper that he published last year, and that's something that you might want to revisit. Hello, Jesse. How are you?
Okay, there's a lot of background noise here, so I probably won't talk much, but hello.
Hi. Right now we're talking about our, a recap of 2020, and I was mentioning earlier that I have this quote about the flow of time, and so it has to do with dinosaurs. Less time separates Homo sapiens from Tyrannosaurus rex, and separated T-Rex from Stegosaurus. And that, I think, that puts into perspective 2020 in some ways. And right now we're going over a timeline of our meetings that we've had over the last year. So we've had the two meetings, the February and the March meeting, and those are just topics. I mean, there are a lot of different topics in the meetings. If you forgot or if you didn't make a lot of the meetings, I encourage you to go over that list. There's a lot of interesting stuff buried in there. And so where do we go from here with that sort of stuff? I mean, that's something that we have to think about. We also should think about some of the other topics that we visited in maybe later in the year, June and July. We reviewed a lot of papers in the group. And so a couple of those papers involved things like morphogenesis and topological data analysis. And so we did a paper, topological data analysis for developmental biology. We also covered some papers on beak and wing shape models. And so those are things that, I like to cover to keep our group current, but also to give people ideas for things that they might want to do. Also in June, I gave a talk on Open Worm to A-Life 2020. So the Artificial Life 2020 conference, I gave a talk at one of the workshops for young researchers, and I prepared a 5-minute talk like the day of, and it was pretty good, and I presented it in this meeting. So I think it's a good recap of the state of OpenWorm. And then later on in the year, we did a lot of other things. We started in September. OpenWorm had their annual meeting, and so during that meeting, I reviewed a lot of the things that were going on in our group for the last year and a half, because we hadn't had a meeting in like a year and a half. So I gave a recap of a lot of things that were going on in the meetings, going on in terms of publications. And so they're very impressed with that. And I think it's jolted some people in the organization in terms of their awareness for DevoWorm. So that's good. In October, we had a Hacktoberfest, which was, we hadn't done it before, really. So we didn't really invest a lot of effort into it. We just kind of, we did some advertising of it. And we got about maybe 15 participants, which is good. Some of the bigger organizations get more participants. But the Hacktoberfest thing, I think, is maybe something we revisit this year with a little bit more effort into it, or, with some clear goals, because we just kind of stumbled into it, I think, this year. But I think it's all right. I don't think you need to have 1,000 people contributing for it to be successful. But I think especially for the DevoLearn platform that we had developed, I didn't put this on the list, but around the same time we released this DevoLearn platform, which involved the different Google Summer of Code projects, and I'll talk about those in a minute. But yeah, we released it around here, and then we got into Hacktoberfest almost immediately. So I think next year maybe we'll have some clearer goals in terms of what that is. And then in November, we covered various topics: developmental brains and meaning, torch dreams, DevoWorm ML updates. All of these are really interesting because they're kind of like fragments of things that people are doing, like torch dreams. We're updating DevoWorm ML, which happened in 2019. And then we have this developmental brains and meaning. These again are papers, on different topics that we wanted to cover and sort of form themes around them. So I have something in the chat here. Okay. Yeah, Ujjwal says 1,000 people contributing will only create mess, to be honest. That's probably true, yeah. I mean, unless you have the infrastructure for dealing with that many contributors, it's hard to sort out. So yeah, I agree. Probably in this case, like growing Hacktoberfest would probably be in the order of 100. But yeah, I mean, we can plan that out a bit more. And I think, the important thing is that people become aware of it and that they're, excited by it. And certainly this year we talked about, or we've been kind of working on this DevoLearn paper. So I'm not going to cover that in this slide, but let me get back into the slides here. So those are the things that happened during this year. And there are many more things that happened. I didn't put them on here. But I just wanted to give people a taste, reminders of what we have done. So here's a more sort of concentrated list of things in terms of lectures. There were actually a lot of lectures this year because we have been, virtual a lot of the year. So that sort of facilitated these lectures. So in April 2020, we had a lecture, Processes Connectivity Towards Biologically Specific Complex Networks. This was at the NeuroMatch 1 conference, and this is a follow-up on some talks that had been given at conferences like NetSci, and there was a paper published in Biosystems in 2018 on this idea of embryo networks. And so we're kind of following up on this, and this is one of the follow-ups to it. In May 2020, I gave a talk at the Virtual Worm Lecture Series, which was a lecture series that followed up on, or that was sort of built in the wake of the pandemic it allowed C. elegans researchers to give talks, whereas the public lectures were canceled. Oh, Arnab is here. Hi, Arnab. Welcome. I haven't seen you in a while.
Yeah, it's been a long time.
So we're talking about our things that we did last year in the group. So that was an interesting talk. Also in July, we did, as I told you before.
I mean, so Bradley, don't let me interrupt you. I mean, you can continue with the discussion that you were having.
Oh, yeah.
Sorry, I don't mean to interrupt. Yeah.
That's okay. So in July 2020, I did this, like I said, I did this talk on OpenWorm at the Artificial Life 2020 conference, OpenWorm Model Realism and the Truth. And this was a cute little talk on like how there's all this hype around OpenWorm about downloading brains into worms. And, I wanted to give people, more general audience, it sort of debunks that's an easy thing, or that you're downloading a brain into a worm, and the way that it's been hyped. And, then it goes into like, what OpenWorm has in terms of modeling, what kind of types of modeling that are involved in OpenWorm. And so if you're interested in modeling or in OpenWorm, you might revisit that talk. I think it's on the list that I told you about. So that's available. Then in September, of course, I did the DevoWorm 2020 update at the OpenWorm annual meeting. And again, like I said, I think I got a lot of people sort of looking again at DevoWorm, because I think like a lot of people in OpenWorm don't realize what's going on in DevoWorm. And so hopefully, that's something that people are paying attention to now. And certainly it was impressive. They said it was impressive, the kinds of things that we were doing here. So that's always good to hear. And there are things that we're following up on from that meeting in terms of education. And so, like we were talking about, especially with like the machine learning education, how do we make that more formalized? Can you make like a machine learning course that sort of focuses on what's going on in OpenWorm, but also make it like a skill building exercise for people so they can skill up in certain areas with, OpenWorm is sort of the theme. So, we did the DevoWorm ML series in 2019, and that involved a lot of different topics, but we always came back to sort of the worm and biology. And so that's something we might be doing in the next year. And then finally in October 2020, we did the NeuroMatch 3 conference, which we had 2 talks from DevoWorm related things and people. So the first was the contrast between biological and artificial neural networks. This is the BNNs versus ANNs, or BNNs and ANNs. This was by Krishna Katyal and myself. We worked on this presentation. So basically the idea was to talk about biological neural networks, which are, networks of neurons in the brain, and then artificial neural networks, which are, of course, computational networks in a computer. And so we talked about the differences between them, talked about maybe like some of the properties of neural networks that we can exploit that are more biological in nature. And, people have been kind of thinking about this more recently. There have been some papers published on it. I think we covered those papers in the talk. So that's something, if you want to revisit it, it's actually in a blog post. So, if you want to, I can repost the blog post in the Slack channel. And that blog post has everything that was presented at NeuroMatch 3 with the video link and the slides and everything. So the second talk then was the Psychophysics of Non-Renal Cognition, which was something that we're doing with the Bacillary we're focusing on these diatoms, this diatom genus called Bacillary, and we're looking at how they move in the water column because they're diatoms, they're marine organisms. And so we're looking at actually like the psychophysics of these organisms. How do they move, and maybe how do stimuli in the environment affect their movement? So that was a talk that actually went pretty well. I thought that it was well received. It was in one of this molecular and cellular biology tracks of Neuromatch, but I think it went pretty well because I think people, I thought it might be a bit controversial, but it wasn't that controversial, so that's good. And so this year we're going to focus on putting out a paper on this topic, the psychophysics of non-neuronal cognition. And we're actually working on an abstract on this topic, contrast between biological and artificial neural networks. And I'm not sure, I was going to submit it to a workshop on the 6th, but I decided not to for a number of reasons. But I think I'm going to submit it to something in March. I'm going to submit it to the SIAM conference, which is the Applied Math Conference. And it's got potentially a much broader audience, and it's a much better chance of getting into the conference. So that, we'll wait until closer to the deadline to sort of get that all in order and submit it. But I think that'll be good. That'll help us follow up on some of these topics that we did public lectures on in 2020. Publication-wise, we have this Towards the Digital Diatom. So this was published in Diatom Gliding Motility. This is in a book that was actually accepted into the book in May. We've had a preprint out on this all year. So we published that preprint in December of 2019. Hi, Mainak. And so that's going to be, that book actually will be coming out this year, but that's one acceptance that we got. Another one was a paper that I wrote on raising the connectome, the emergence of neuronal activity and behavior in C. elegans. This was in Frontiers in Cellular Neuroscience. That's another follow-up on some of the work that we've been doing on the developmental connectome. And then this paper, Data Theoretic Synthesis of the Early Developmental Process. In December 2020, this was accepted in Neuroinformatics in their special issue Neurocommons. So this was a paper that came out of it originated in a call for papers by the OpenWorm Foundation. OpenWorm was going to publish a special issue in the Royal Society proceedings. And so we wrote a paper. They didn't want it because it wasn't really, within the scope of what they were doing as much. We ended up putting some of that material into the main OpenWorm paper in that issue. And then this paper kind of got spun off, and it was rewritten. But then now it's accepted in Neuroinformatics. So that's another, that should be coming out soon. I just did the proofs for it this weekend, so it shouldn't be too long before it's out. And then this new paper, Periodicity in the Embryo, which, again, this is something Ujjwal and I are authors on. And this is out in bioRxiv today, or Friday. I didn't add the link to that. And it's also submitted to Biosystems. So we had a lot of action.
bioRxiv is where the pre-published state of the paper is there, right?
Yes.
In review.
Yeah, it's a preprint version. So, we'll probably update it, with a new version if we have comments from reviewers. But I actually can put the chat in the link in a minute. And so then that's good. We can follow up on those publications. Another thing I didn't add, because it isn't really out yet in any way, is a publication on the DevoWorm materials. So we did Google Summer of Code 2020, and we had Ujjwal and Mayuk. and they were our students for last year. And they worked on different Mayuk worked on this project DevoWorm, which was a pre-trained model for microscopy data in development. And it came out very well. He did a very good job. He set up this PyP repository for the software. Ujjwal did this DevoWorm AI project. So he worked on bringing together a lot of the stuff that we had in terms of AI, in terms of our... we have this thing called DevoZoo that has a lot of educational materials. So just bringing that all together. And so they both did very well in their projects. And we decided to build a GitHub organization called DevoLearn. So this is the software DevoLearn. And then there's a broader GitHub organization called DevoLearn. And DevoLearn it contains this pre-trained model, this DevoWorm AI initiative, also some other files on like data science tutorials, some other educational materials, and we're building it out in a number of different areas. Now it's still very early days for it, so we released this thing after Summer of Code, which was in September, and then in October we sponsored a Hacktoberfest, so people could contribute to the DevoLearn platform through Hacktoberfest. And we got about 15 people to apply and to participate in Hacktoberfest, which is, a fair number. We could have more people. Of course, as Ujjwal mentioned, if you scale it up too much, we really do. It is messy. You do need to have a lot of people dedicated to it. So, we might want to grow it this coming year, but maybe not as much as, some other organizations that have more attention paid to it. So that's something to think about. So this is something that we did, Google Summer of Code. It resulted in DevoLearn, and now, we've had public events based on DevoLearn, but we'll also have a paper coming out on DevoLearn sometime early this year. We already worked out a paper for another venue that didn't happen, so that paper is just going to be continuing on, and I hope to get it like in the format of where we have all the different components of the DevoLearn platform and kind of give a good description, and then release it as a preprint as a good first draft, and then we can maybe publish it from there. So I think that's moving along well. This is DevoLearn. This is actually the launch, September 2020. This is our logo. The organization is on GitHub. It's github.com/DevoLearn. Please visit and check it out if you haven't already. This is our weekly meeting YouTube archive. This is another thing we did in 2020. And this is, I look back at this and I say, we did a lot of stuff in our meetings. And we have a lot of video for it. So now the question is like, how can we follow up on some of this stuff? Because again, like in the first slides, I mentioned that we were going to have like, we had all these things that we did, different points in the year, and it kind of blurs together by the end of the year. So, maybe we should go back through these talks and look at like some of the things that were mentioned, some of the papers, some of the techniques, and really kind of think about, maybe pick up on some theme and bring it forward into 2021. But we do have this YouTube archive, so I think it's very valuable to have prior meetings up on YouTube so people can engage asynchronously if they miss the meeting. We can also do topic mining, again, like I said, mining these talks for different themes and other things. So that's all I had to say about that. Any comments about this coming year, the past year? I know Arnab and Mainak haven't been here for most of the past year, although Jesse and Ujjwal have. So I don't know. Maybe they don't. Maybe they found something that they didn't know was going on.
So I think Jesse has written something in the chat.
Okay. All right, here we go. Let's see. Yeah, let's go to the chat. Working on it. I know it is the wait a bit. We'll submit soon. That's, I think, on the one of the papers, or I mean, we're always JOS. The JOS paper is now just something that we're doing as a preprint. Like the JOS people thought, we weren't really, it wasn't very important yet in terms of software. So they're looking for, I think, scientific software with a pretty big user base. So, but we'll be working on that paper for a preprint. So yeah, we'll just call it the JOS paper as a shorthand. Jesse says, I'm curious what papers are in development now or can be contributing to. Yeah, so I mean, I think we mentioned this about, let me see if I have it here. We have a place in the, if you go to the DevoWorm GitHub and you go to the group meetings, there's a list of open papers. And so let me see if I can do this. I wanted to talk about these public lectures, but I will go back to that in a minute. So if we go to the DevoWorm GitHub and we go to group meetings, there's the README and group meetings, and this is where we have a lot of the open papers. And again, this isn't updated. I need to update it more. Some of this stuff is like kind of outdated, but these are the open papers. I probably will put in something on the DevoLearn paper, although that's just really kind of the people who were involved in setting up DevoLearn, but people can contribute if they want. Maybe I should have a little bit better list of these, but these are the group papers for now. There are also papers going on with respect to the I know Jesse knows about the I have another spreadsheet in another place where we have submissions that are going to be made, and I'll put those up on this README. Basically, we have upcoming papers and upcoming venues, and I put it in a document that's I don't have right now open. I can actually share it with the group, maybe in the OpenWorm or in the DevoWorm channel in the OpenWorm Slack as well. We have some things that are going on that aren't on the open papers list, like there is this abstract on sort of developmental biology and machine learning and deep learning, but that's something that, again, I'll probably just put in that. I'll put in that list, and maybe I'll make it open at some point. But yeah, I think if you're interested, maybe we should talk on Slack about it. Like, what paper specifically are you interested in contributing? Because contributing is a kind of a investment of time. It's like deciding what's the best place to contribute and what exactly you want to contribute to it. Otherwise, it's kind of like you never really get around to it because it just gets overwhelming. But and again, it's like things that are, sometimes there are papers that have been sitting there for a while and they just need some focused contribution to push it forward. So that's another thing is, so I'll go back over this list again and try to update it. But that's where it is. It's in this group meetings README. All right. The other thing I wanted to also put a link to is this preprint, so just to make sure it's in the chat. This is the periodicity in the embryo preprint. You were going to say something, Jesse?
Oh, no, I don't know. Okay, I think you can hear me okay, right?
Yeah.
No, I was just going to say, I was going to mention the periodicity paper, and I saw that I will follow up something in Slack, and I had been looking at the paper. I didn't really push any of my contributions to the doc, so it's fine. But I at some point I might go through these things and sort of like what we're talking, like not to go full lab manager as I would in the other group here, but like organizing some of the papers and the contributions might be like a personal project that I get into at some point. But like, you know, not excessively to do it, but just at least just to review for myself. I'm going to go through those things again. I know I mentioned it before, but I'm kind of like, all right, I have the time to do it now, so I'll do it that way.
Yeah, the periodicity paper is like, this is the first draft, so we'll be getting reviewer comments back, so there's still time to contribute to that. It's just a matter of, like, you could become an author on it in a later version, so that's not like impossible. It's just that, like, we get it to a certain point, we put it into a preprint form, and then we'll have different versions, we'll update it, and then eventually, if it's in a publication, then that's considered sort of the end of the road for that. And then if we want to do something more from that, we can write a new paper and start over. So it's that sort of iterative process where we build up to this publication, and then we maybe, if we want to we find something that's interesting, we might start a new publication from that. Or, even if it's something that doesn't fit into an existing paper, it's something we could follow up on separately. That's how a lot of things start. You get, like, they butt off, you think, well, this is a good kernel of an idea. Let's do a paper on this, or let's do a presentation. They kind of come out of these ideas. So, I've written paper outlines before, or papers where they just they go all over the place. They don't really work, but we'll take a couple of ideas out of it and, like, develop them, and then they end up being the papers that are actually published or pre-printed or whatever. So, like, the stuff, I've done a lot of stuff with, we've done a lot of stuff earlier in DevoWorm where, we were batting around ideas and developing papers, and then, there'd be some ideas that would come out of it, like the networks, the embryo network stuff. That kind of came out of sort of applying network analysis to, development and thinking about it, and then eventually we came up with that idea. So, it's don't worry about, like, if it really contributes to a specific paper or not. If you have a good idea, it's good to get it out there, even if it's a separate thing. If it's a separate paper, it's fine.
Yeah, that makes a lot of sense.
Yeah. So that's good. Does anyone have any more questions about that or comments?
So Bradley, I'm sure you have your hands full right now because of so many different things that are going on right now. You presented a lot of different threads that you're sort of focusing on. So I'm sure you read the email that Malin had sent about soliciting ideas for GSoC 2021. So yeah, I just thought I'd jump into this call and just see how things are. Of course, I've not been around for a long time, so I see that a lot of new exciting work is happening.
Yeah.
So have you maybe started thinking about GSoC 2021 ideas?
Oh yeah.
And also, importantly, this time I think the project length has been slightly reduced to, I think, 10 weeks, if I'm not wrong.
That's what I understand too, right? Yeah, it's 10 weeks. Actually, that's what we're going to do next. Let me share my screen. We've solicited a couple of ideas here from people over the holiday. And again, so thank you, Arnab, for attending. And yes, so we have coming up, I think, this week, INCF, mail-in from INCF. She wants these descriptions, but she doesn't need like a full formal description. She just needs the ideas, I believe.
Yeah.
So this is our GSoC 2021 repository. So we have 3 project ideas that I put up here in the GitHub repository. And so we'll go through these one by one, and we'll kind of talk about them. And if we have ideas about them, we can talk about them here. We can, issue a pull request to edit them. I'm going to gather them up this week and send Malin a description of each of these. And then I think by the end of the month, we need to have formal proposals that are polished, one-page descriptions, and there's something that we can then show to potential students. So the first one is by Mainak, and this is upgrading DevoWorm. So this is the DevoWorm platform that I talked about, the GitHub organization. So this is so there's the DevoWorm software itself, and then there's the platform. And I think he's interested in maybe more of the platform, but we can also, there are other components to this for them to work on as well. So there's the DevoWorm pre-trained model. There's also another C. elegans specific platform that was the product of an earlier GSoC. There's some other software that we're developing in there in terms of we have other programs that we're interested in. So these are all kind of fall under that umbrella. There would be 4 key elements in this project: improving the current models, training and adding more useful models. So as I said, we have this group of models already that we maybe want to improve upon or add new models or modify them. Improving usability, which is always a good thing. And interactive online demos, which would be an optional thing. Now with 10 weeks, of course, we'd probably have to pick one of these elements. Although there'd be if we picked improving the current models, we could say training and adding more useful models might be you would work with improving some of the models and then proposing next steps or something like that. Improving the current models, we have the basically it's like benchmarking these models, so using a number of methods for that. So that's one thing that could be done. Adding more models. So DevoWorm should also contain pre-trained deep learning models from other species, which are of high importance in developmental biology. We can split up the different species so he gives an example here in pseudocode for DevoWorm C. elegans, for DevoWorm zebrafish. You just have these different models specialized for different species. And improving usability. As the library grows, we should have a better place to host proper documentation on a static website, which could be named like a docs website. You could use a wiki as well. We could use a lot of different platforms, but basically to have a formal docs section and online demos. So the online demos are something that Mainak did last year very effectively. He had a number of Colab notebooks, and I see that Mainak actually has created a Colab notebook as well on some of the stuff. He's been doing that as well. So these are very good. I like Colab notebooks because they give people nice demos in a very, usable space. People can just go to the Colab notebook and run it. But they're still a bit intimidating for people from non-CS backgrounds. So we should focus on making interactive demos. So we talked about this a bit, I think in the fall, where we talked about we were looking at some of the agent-based modeling platforms. So your interactive demos would contain slider-like mechanisms to interpolate between different values. So it gives it a link to a distill publication. So yeah, that's one project. Now that's Mainak proposed that. Now, if you have ideas, you can issue a pull request, maybe ways that we can make this smaller, because this has to be fit within 10 weeks. So this is a lot of stuff here. And this could be 3 or 4 projects, but we'll probably distill this down to maybe one, and then, maybe we'll get people involved in other things. So the second project then is this Bacillary and non-neuronal processes. And this is something that Ujjwal proposed. This is something that Ujjwal and Arnab Singh worked on, I think, in 2019. They did this thing in the summer. It wasn't part of Summer of Code, but it was a project where they worked on applying a deep learning model to these bacillary images. And so they're creating digital simulations. The idea here is to add more functionality to that original project. You're thinking about maybe a wider range of deep learning approaches. We also need to add this functionality to the DevoWorm library and web tool. So this is again expanding the number of species that you have in the DevoWorm platform. And then students will be required to do some literature review to get on board with the project. It'll be a great opportunity to learn and develop a cool project. We need documentation. We need people to do, use Colab or Jupyter notebooks. And, this is just the description. And then the idea would be to create sort of an online GUI that would execute this model. And so that's the vision there. I'm going to edit it some more, and we'll probably also submit this one. I think this one might be, if we can get down the goals of what we want them to do in 10 weeks. I think this is going to be a good one to do as well. Do you have any comments on that, Ujjwal? Well, Susan? Okay, so let's see. So Ujjwal says, I was thinking of using POS tagging mechanism on genes, reading more about it. We'll post rough outline soon. Okay, I think that's from earlier. This is the thing you were talking about. Yeah, I think we were talking about this in Slack. Okay, so Ujjwal says, I can give weekly goals for the project. Yeah, that would be good. Well, if we could have some sort of if we could take this proposal, Ujjwal, and break it down into maybe what we might expect over every 2 or 3 weeks. What do we expect from them? And then we can get it. Because, this week I just need to know I just need a short description to send to Malin. But I think for the final thing that we're going to give to students, I think it would be good to have it's always good to give them kind of a rough outline, because they have to build those schedules. They have to schedule their time, and that's always one of the hardest parts of the proposal, is making a timeline. So if we can help them with that, that's good.
Sorry, can you hear me?
Yeah, I can hear both of you. Yeah.
Okay. Oh, this off.
Am I audible?
Yeah.
Yes, so I will surely update the proposal. Again, just a rough idea what I was trying to say. So I will surely update this proposal this week, give the clear goals, what has to be done in this project. We have clear goals, actual methods.
Yeah, I think it's good. I think it's a pretty good start, though. I think it's definitely something that would be interesting. The one thing we did talk about also is that we need a neuronal part, or a brain part of this, because it's INCF. I think, though, that it's I think I can add that in also. It's it's always they've been interested in DevoWorm's projects, and they've always been kind of tenuous in that way, because we're not real. We're OpenWorm, and we're also in C. elegans. You're also segmenting neuronal cells, but we're doing developmental stuff. So I'll just put a statement in about how it's sort of related to the brain. And I think this one is from Krishna. This is DevoNet, Specialized Convolutional Neural Networks for Biological Data. So Krishna mentioned this. I think he presented on this in the fall, on this idea of a biological general model. So he's interested in this idea of taking images from as many sources as possible and extracting features, which is really hard to kind of do. A short. You have to get your arms around it first before you can really get anywhere. And so this one this is a sort of also a pre-trained model. And I think the idea here is to have a pre-trained model for a lot of different biological images, like, not just microscopy of different organisms, like, invertebrates and vertebrates, and, but also to have like plants, I think, and X-rays from, humans and other sort of neuroimaging from humans, which is really, I think, maybe a bit too ambitious, but the idea is really different than what most people were doing, like at the species-specific level. Again, we haven't really done anything with this, so it might not make any sense to go that broadly. Maybe it makes more sense just to look at, across species instead of within species, because the models that we have now are really kind of specialized for specific species. That's the way we kind of train them. So their performance is best maybe with one species or another. In this case, it would have something that would be cross-species. And so he kind of lays out the description of this. And this is, again, something we need to work on a bit more. But this is another idea. So we have those three ideas. And I think that those are probably pretty good. We'll probably do like three. We'll propose three projects. And then, we will, maybe get students for all of them, maybe for one of them, maybe even for none of them. I don't know what the situation is this year. We usually get a lot of interest in the machine learning projects. So it's not, it's never really for a lack of students. We'll get, a number of applicants for each project, hopefully, and then INCF will make a decision as to which ones get accepted, and then that'll be. But I think it's always good to have like more ideas than fewer ideas, because then we can like really get them down to something really good. And we'll be working on that. I'll submit like a short description of each project this week, and then by the end of the month, we'll have to work on refining it a bit more.
How about a project for making a shell out of 3D images of a sphere?
Oh.
I'm coming up with some more different images of, canola seed at the moment, or peppercorns. Instead of salamander eggs, but I'll be imaging salamander eggs too if I can get this system to work.
Oh, wow. Well, that sounds pretty good.
Yeah. It'll be 9 images, hopefully, around the sphere.
Okay.
In fixed positions. So that might be easier to work with.
Right. Yeah. And then the idea would be to make like a sort of a digital sphere or a way to sort of view them continuously.
Yeah. I mean, you could view each image in a montage, but it would be better if you could somehow make this into a sphere and then follow what happens with cell behavior as you image it.
Yeah. So that's something that I hadn't thought of for Summer of Code. I mean, we may be able to either incorporate that into one of them or use that as another idea. But again, like I don't know what again, we have to kind of, for INCF at least, we have to have like a neural angle to it. But,
there is. There is.
So you're wanting to look at
neural plate development into the brain, how it rolls up into the brain. I hope I can do some mechanical simulations of part of it myself. So that might add to it, but I don't have time to do this. Like, I don't have time to figure out how to make a sphere out of 9 flat images.
Yeah.
From all sides, I guess.
Yeah. Well, it's definitely something. Well, let me write. Actually, I could write that up myself, and then we could actually submit that as well. So that's another one. And then that's something we can do like outside of GSoC as well. But thanks. Yeah, that's good. That sounds good.
Okay, it would be good for a number of things,
Right.
including satellite images.
Yeah, it would. Yeah. So, okay, so that's the GSoC update, and hopefully, by the end of the week, I'll be sending it to Mail, and then I'll send everyone a sort of an update on what those look like. So let's see. All right, so that's that, and then finally, I'd like to wrap up here with a couple of things. First of all, Mainak, he put a message in Slack about improving upon the DevoWorm code base. He was talking about how the generative model generates 60 by 64 images, which can be interpolated to higher resolution. But then, he's talking about making them higher resolution, and then he's going to issue a pull request on this. So, Mainak, is there any updates on that?
Yeah, Bradley, I've been working on it for the past couple of days. So yeah, I'll issue the pull request very soon. I'm actually testing the library right now in the virtual environment. So after everything is done, and then I'll send the pull request. Yeah.
That sounds good. Yeah, I look forward to seeing it. So.
And yeah, as you said, right? It currently only generates 64 by 64 images. So what's happening is that it's actually interpolating the images to a higher resolution, which is actually leading to a loss of data, right? The finer details are getting lost out. So what I'm basically doing is that the new model will actually generate the high resolution images itself. It won't have to be interpolated by that scale. So there will be a lower loss of data.
Right.
So yeah, that's the reason.
Well, that sounds good. Yeah, that sounds good. Look forward to the pull request. We also have 3 papers here. I'm going to go through them really quickly. If you have to leave, that's okay. But I just wanted to go through these things that I found here. Or actually, the first one we'll go through here is this is something that Susan sent me. This is from Quantum Magazine, and it's called A Newfound Source of Cellular Order in the Chemistry of Life. Susan, what are your thoughts on this?
Cells are packed. They're very closely packed, and that has implications for how they function and, of course, their chemistry. And I heard a lecture about this back in 2005, I believe, and it's related to how the cytoplasm moves and how cells move. They're what you call thixotropic. They're like catsup, and you shake it and shake it and shake it upside down to try to get your catsup out of the bottle, and all of a sudden it whoas, and cells are kind of like that, and this is why they're very tightly packed. So this was a nice summary article in more or less English, so that hopefully people can read it. Anyway, it goes into more detail than what I just said, and the nucleus is like this as well. So
this is a picture of the nucleolus. The largest structure inside the nucleus is a condensate with internal structure. And so in the ST nuclei from frog cells, condensates of different proteins nest with one another. So a condensate is a sort of a physical property of something where there's I can't remember the definition of a condensate, but
it's okay.
Oh, yeah. So
I
that's
should have looked that up for you.
yeah. No, I mean I've heard of it. I just don't. I can't give a really good definition right now. But
Well, if water condensates from a gas to a liquid, well, it's consolidating its molecule. So this is something similar, but it's going from a liquid to an alternate, a dense liquid. I don't know. That's my guess.
that sounds good to me.
Like
Anyways,
I say,
yeah. So I mean, these quanta articles, they have a narrative, so it's pretty easy to follow along, and so they were talking about let's see, they were talking about the phase separation in cells trickled out around 2012. I wonder if this Hyman is the same person who did there's a method that I used to use for getting different fractions of RNA out of a cell. I don't know if this is the same person, but it sounds like that's what maybe they're trying to do here. They're trying to like figure out different parts of the phase separating different parts of the cell out, and so biomolecular condensates in cells. In complex cells, phase separation drives proteins and other molecules to aggregate into fluid bodies, or condensates, with diverse functions. A few of these condensates are shown here. So this is different things like nuclear speckles, the nucleolus, histone locus body, DNA damage body, and all these different things in the cell that are sort of aggregated together. And so, this of course, if you know anything about the emergence of multicellularity, there's one hypothesis that says that a lot of the multicellular cells, say you see in humans, come from like smaller cells, like eukaryotic or prokaryotic cells, which are just very simple cells, and then other things that were in the environment, and they were all kind of came together into this new type of cell, which was the eukaryotic cell. And so, I mean, I don't know if they don't talk about this here, but it's kind of an interesting thought. And so they go into what drives droplet formation here. Condensates and gene expression. So condensates seem to be involved in many aspects of cellular biology, but gene expression and the production of proteins is one area. They talk about the ribosome. So this is, there's some interesting stuff there. The ribosome, of course, is where RNAs get translated into amino acids, which then build proteins. So that's an interesting system. And also, if we have time in another meeting, we can talk about the ribosome, because it's a really fascinating system, and it does a lot of things that go from like gene expression to protein production. And you think about it as just being genes to proteins, but there's a whole other layer there in the ribosome that really makes a difference. Actually, that's very similar to what I was thinking. When this method that I was talking about, they would take different fractions of the RNA and they would take it off of the ribosome. So it's like isolating the ribosome, and in isolating the ribosome, you can get a lot of the translational RNA. And that's different from a lot of the transcriptional RNA, and that it's been, selected for by the ribosome, and it basically gives you an account of what's going on with gene expression versus protein production. So that's,
if people want to see this in cartoon form, there's something called the virtual cell, and it's the University of North Dakota. Made some very nice biology movies, and transcription and translation are two that are very nice to watch. It kind of gives you an idea of that system.
Yeah. Well, and
North Dakota State University. Sorry.
Oh, yeah. North Dakota State. Okay. Yeah, it's very good. Yeah, this stuff is like very hard to understand sometimes without the visualizations. But, this paper. Yeah, this gives a number of links to different papers here. where they talk about a lot of these different things. So this is a good paper to follow up on, and if you're really interested in like the physics of a cell, it's really fascinating how condensates pack genetic material. So they talk about chromatin and how chromatin get packed in, which is another interesting problem because there's a chromatin, it's like where the DNA is hosted, and it's there's a lot of it in the cell, but it gets packed into a very small area, and so that's another thing that you might be interested in. So all these things too, they have like, if you're interested in machine learning, I mean, there's a lot of that if we can get images, and people are producing high resolution images. They've also produced, of these different parts of the cell. There's a lot of EM or electron microscopy data out there that might actually be sort of fodder for machine learning in that. But that's a very interesting article. Thank you, Susan, for that.
Okay, put the virtual cell in the chat.
Oh, okay. So that would be this link here. vCell NDSU NODAC edu. They're animations. So check that out. And then I wanted to talk about another paper related to this, which is the microscopy, continuing microscopy, something called expansion microscopy, and this is expansion microscopy of C. elegans. And this is a new paper in eLife, and this is Edward Boyden, who's at MIT, and he developed this technique called expansion microscopy, and this paper is specific to C. elegans. So they've developed this technique called expansion microscopy, where they basically take the sample and they swell the sample up. They swell it out so that you can see things that are very small resolution, and they expand it out with some chemically processed tissue or hydrogel embedded tissue. So they take tissue, they embed it with this hydrogel, and they expand the tissue out. So it's like one way to get at very small structures. I mean, one way is to get really high resolution microscopy. Another way is to expand things out. And so, at least as I understand it, I'm not an expert at this. But they've done this in C. elegans specifically. So this is the paper describing this technique applied to C. elegans. So so recently we discovered that it is possible to isotropically expand biological specimens by permeating them evenly and densely with a swellable hydrogel polymer network, anchoring key biomolecules or labels to the hydrogel, softening the tissue through a chemical process, and then adding water, which swells the polymer and in turn the tissue. So they hydrate the sample afterwards and it expands it out. This technique, expansion microscopy, EXM, is now being adapted and improved by many groups and has been applied to the tissue of mice, human patients, and in many other biological contexts, and there are a bunch of references. However, C. elegans is wrapped in a multilayer cuticle, which is well known to be impermeable to many small molecules and antibodies, and mechanically stiff to the point where physical expansion would be expected to proceed poorly. So thus we set out to develop this protocol customized for C. elegans. So they had to work out a custom protocol for this because of C. elegans. Biology makes it hard to do this. So they did this. Let's see if they have any images. This is a diagram of what they did for the protocol. And then I wondered if they actually had any microscopy images in here. Okay, this is Excel enables antibody-mediated visualization of fluorescent proteins. So these are the fluorescent proteins. So these are proteins that have a fluorescent molecule attached, and they're usually bind to some, specific target in the cells. So this is, this is where you might get like a specific gene that's expressed or protein that's present somewhere, and you'll put a fluorescent molecule on this reporter, and then it'll glow, I guess is the correct or sort of a popular term, in places where that is bound to the that fluorescent molecule ends up bound to the target. So you can see, like in this area with red, you have a lot of this fluorescent molecule bound to its target. In the places where it's not red, it's not. And they show like kind of examples of this where they're able to do this with different methods. So this is, it's one example. I don't know if you can see it very well. So this is another one where this is a better actually example. You have larval stages 2 through 4, or 2, 4, and then day 1 adult. So these are different. This is as the cell is developing post-embryonically. You have pre-excel, which is before they implement the method, post-excel, where it's expanded by 3.3 times its original size, and then they're doing this registration technique where they're overlaying the pre and post-excel images. And so here where they have like basically a worm where they haven't applied this technique, then they have applied the technique, and then they're registering the image so that they can, figure out where things are in the image. And so that's an example there. So it's a lot of technical detail. Go ahead.
Do they put distilled water into the gel, and then that causes the worm to expand? I've accidentally done this with a salamander embryo, and was very upset with myself because it died. That would also cause this to die, does it?
I think it does. I don't think you can do it in living tissues. I'm not really sure. I think they've worked out, I think they're fixed tissues. I'm not sure. I've read about this a little bit, so, but I don't know. But yeah, I think you can't do it in vivo. You have to.
They killed the salamander when I put it in the distilled water for a while.
But I think the idea is they can do this in a controlled manner, because you can imagine, if you expand a bunch of cells, if you freeze an organism and you unfreeze it, a lot of times there's a lot of tissue damage. Like in C. elegans specimens, if you freeze down, you can freeze down C. elegans, you can freeze them in a solution and then bring them back, hydrate them from, they thaw them out and hydrate them, and there'll be some eggs that survive, and but there's a lot of tissue damage to the worms that were already in that sample. So you have to do this in a controlled manner, I guess is my point. And so this is showing kind of how this method reveals anatomical structures. So it's some of these anatomical structures are very clear, much clearer than you would get with something like a standard microscopy technique. But there is this electron microscopy technique that's better, but it's harder to use. And so this is just using a standard microscopy setup, but you're actually getting a higher resolution of things. I don't know how it compares to electron microscopy, but these are pretty good images, so I don't know. So yeah, this paper, again, if you want to read this paper, it's a protocol, so it's probably going to be hard to read for many of you, but just to go through the images, just to get an appreciation of the technique. So Jesse says, I know Logan Thrasher Collins is into this stuff and Ed's work. If there's a particular need, I could ask him to come to the group and talk sometime. Yeah, that'd actually be good if you could set that up. That would be a good talk to have. I think there would be a lot of interest in it. So yeah, I know that. So Ed Boyden does a lot of stuff, a lot of innovative things. So, but that would be good to have if we could get an expansion microscopy talk. I think it's an interesting technique. And then finally, I wanted to talk about one more paper before we go. This is a paper that. So this is something I just. This is something from 2009, but I think it's, something I ran across. And actually, I wasn't aware of it, but these are about the first embryos. And so this is called Complex Embryos Displaying Bilateral Characters from Precambrian Doshantuo phosphate deposits in China. And so this is, they do a lot of interesting paleontology in China. And so this is one of the examples. They do a lot of stuff with like the origin of flowers and things like that. And one of the things they found is they've actually found from very early Precambrian embryos. And so they say here, three-dimensionally preserved embryos from the Precambrian Eukaryotic formation has attracted great attention as the oldest fossil evidence yet found for multicellular animal life on Earth. Many embryos are in early cleavage stage, and most of them yield a limited phylogenetic signal, which means that they find embryos, but they don't exactly know where they fit into the tree of life. So this is just a report. This is a standard, like, paleontological report on what they've found. And so here we report the discovery of two embryos that are three-dimensionally preserved and complex. Imaging techniques using propagation phase contrast-based synchrotron radiation. I think we've talked about using synchrotrons before in the group for imaging things. Reveal that the organization of cells demonstrates several bilateral features, including the formation of anterior, posterior, dorsal, ventral, and right-left polarities, and cell differentiation. Unexpectedly, our observations show noticeable difference in organization patterns between the embryos, suggesting that they represent two distinct taxa. That means that they're of two different species, and so they're comparing embryos, and they're saying that there is more than one species in this assemblage. These embryos provide further evidence for the presence of bilateral animals in this biota, which is this group of things that they've found. Furthermore, these bilaterians have already diverged into the distantly related groups at least 40 million years before the Cambrian radiation, which is a time in evolution where you start to get a lot of different species, like the birth of sort of vertebrate life, fishes, all of that come in the Cambrian, or in this radiation, indicating that the last common ancestor of the bilaterians lived much earlier than is usually thought. So basically this paper tells us that embryos existed before the Cambrian explosion, which is a time when we thought that like a lot of vertebrate life originated. And there was, before this time of vast radiation, it's really not clear what was there. We know that there were some microbes and some other organisms that existed back then, but we really didn't know anything about when embryos emerged. And so what they're suggesting is that embryos emerged much earlier than a lot of the life that emerged in the Cambrian explosion. And so this is Precambrian postgastrular embryo. This is the example of the embryo here. So, this is before vertebrates, invertebrates, all of that we know from a lot of the animals that we talk about in the group. But you have these embryos. And so this is an example. This is just imaging of this embryo. This is another example of the embryo. So you can see the distinct cells here in this ball. And so they're interpreting this as an embryo, and they're identifying different key features of the morphology. This is where they give different anatomical views of the embryo. So let's see what they have to say about this.
There, you can put that in your 3D sphere thing.
Yeah. So this actually came from the boring billion work that, like, I was doing some research on the tree for boring billion, and I came up with this paper, and I had no idea that this had existed. I don't know if that means I'm not reading enough or what, but this is a very interesting paper. It kind of goes on about this embryo, and this is really the first embryo that we found. Again, it's, this is a time when you had sort of the emergence of what they call metazoans, which are animals. You don't really have any distinction between, you have bilaterians, which means they have the two sides of the left side and the right side of the organism, which are sort of mirror images So there's that symmetry. But there's really, you have like not much else. You don't really have vertebrates. You don't have invertebrates as we know them today. They're just these bilaterians that exist. And these are like the early versions of these bilaterians. So, yeah, this is an interesting paper. There's a lot of good imaging in here. And again, like Susan said, you could really do some interesting visualization of this. I don't know if they have a data set. That'd be interesting to see if they had like a microscopy data set available. But, that's something for another time. So that's all for today. Thank you for attending. And if you need any, if you want to talk on Slack about anything, please do. Next week we'll be talking about probably more papers and probably the GSoC stuff again. So, and if you want to present anything, please let me know. Thank you for attending. Have a good week.
Take care.
Take care.